So we're group 2. I'm Caitlin, and I'm going to talk to you guys about the introduction of the article that we looked at. After me, Bethany will go and she will talk about the materials used in this study. Then Lindsay will go over the methods that they used for the study. Ryan will go over the final results, and then Kim will discuss what the results mean, and she'll conclude our presentation. So the article we chose was by Richard Wyman, and it was entitled Experimental Assessment of Salamanders as Predators of Detrital Food Webs and Their Effects on Invertebrates Decomposition and the Carbon Cycle. So, in his introduction, Wyman talked about how human activity had most likely led to the extinction and population declines of many different amphibian species across the U.S. Amphibian populations, including salamanders, have continued to decline for many, many years. And while there have been studies that have focused on the reasons for this decline, there haven't been a lot of studies done on what the effect of the decline would be on the ecosystem. So that's what Wyman decided to look at. His big question was, are amphibians important to ecosystem function? And the species he chose to use was redback salamanders. Their scientific name is Plethodon cinereus, and he chose them because they're the most common species of salamander across the northeastern United States, and that was where his study was taking place. And they're also a pretty common species in several different areas across the U.S. And previous studies had suggested that salamanders could also affect the decomposition rate, so that's another reason why he chose to use salamanders. This experiment examined the effects of redback salamanders on invertebrate populations as well as on the decomposition rates of leaf litter. And his final results ended up suggesting that redback salamanders are, in fact, a significant keystone species that plays a really significant role on invertebrate populations and on decomposition processes in general. So next we are going to have Bethany come up and talk to you about the materials that were used as part of this study. So Bethany.
Okay, so Wyman, in his experiment, the materials he used were in 9 enclosures in Hewitt Preserve, which is a deciduous forest regions where the redback salamanders would be. Three of the enclosures had no salamanders. 3 of them had 2 salamanders, and 3 of the enclosures had 6, so you have your total of 9. 1.2 by 2.5 meters each enclosure was, or 3 m squared. Each one was made of wooden frames with aluminum mesh that was 2 millimeters thick. The aluminum mesh was around the enclosures so that mammals couldn't get in and eat the salamanders, and none of the salamanders could leave the enclosure and no new ones could come in. That way the experiment wasn't messed up. There was also a plastic skirt on the enclosures, which that was for the massive horizontal movement of the microinvertebrates, and the microinvertebrates in this study were 1 millimeter in all dimensions. 20 centimeters of the enclosure was buried underground, and 30 of the centimeters of the enclosure was above ground. There was also fiberglass mesh, which was 2 millimeters thick on the tops and the bottoms of each of the 9 enclosures. And now Lindsay is going to come and talk to you about the methods in which Wyman used for his experiment.
Okay, so for the method portion of this experiment, Wyman did a variety of different ways of collecting the invertebrates to test them. First off, when the enclosures were placed into the deciduous forest region, he removed humus in 30 by 20 by 10 centimeter rough cut sections from the area he was going to place the enclosure. Once he had removed that very carefully to make sure he did not disturb the section, he put the enclosure in its place. Once the enclosure was placed there, he then restored the humus and the leaf litter with it and made sure that everything was not disturbed. After that, 4 kilograms of leaf litter was added to the enclosures so that he could later do specific calculations. Along with that leaf litter, there was also 60 mesh bags containing 3 grams of dried litter placed in the enclosures and dispersed throughout them. Now, for this experiment, the invertebrate population had to be tested. So how he did that was bi-weekly for 48-hour periods with sticky traps. This was one of the 4 methods. The sticky traps was a sheet of paper with a sticky residue on top of it. These were placed both inside and outside of the enclosures. The sticky traps inside the enclosures could then be compared with those outside the enclosures to see if the population community size was different between the two, and if the sizes of the invertebrates stuck to the sticky traps was different between the two. Also, between those 60 mesh bags that were dispersed throughout the 9 enclosures, 8 of the mesh bags per enclosure was taken back to the lab and put into Burley's funnels for a duration of 48 hours. This was done so that they could funnel the invertebrates through and see how many invertebrates came through and count them. A 40-watt bulb was used for this procedure. After the experiment, all of the enclosure's contents were also bagged up and taken back to the lab. Of those bags, he had experimenters in the lab look through them randomly without knowing which part of the experiment they were part of, and handpicked through the leaf litter, taking all of the invertebrates out of it and counting them. He also had the same procedure done with the handpicking for the humus section. And what the humus was is the rocky deposits that are dispersed throughout the deciduous forest region that scatter along the forest floors. So, how is he going to actually find his results for this experiment? Well, he had to identify the taxonomic categories of all of the different invertebrate species that he collected throughout this experiment. So, of each of these taxonomic categories that were discovered, he then counted the populations for each of them. Once the populations were determined, he sized each of the invertebrates individually so that later he could do calculations to determine whether or not the salamanders liked specific sizes of invertebrate for their prey. After that, the remaining mesh bags were dried and weighed. This was in order to figure out the decomposition rate during the experiment. The difference of the initial 3 gram of dried leaf litter that was placed in each of these enclosures and the final weight in those mesh bags is what determined the decomposition itself. And now Ryan will talk to you about the results of what these findings had.
Alright. I'm Ryan, and I'm going to be talking to you about the results of this experiment. And to begin as a reference, at the beginning of the experiment, the frequency distributions skewed toward larger invertebrates outside of the enclosures rather than inside the enclosures. And that's meaning that the smaller invertebrates were inside the enclosures. And after the experiment, the frequency distributions of major taxonomic categories of invertebrates collected on sticky traps and did not differ, meaning that the salamanders did not differentiate between different species. The majority of organisms caught on the sticky traps were less than 0.1 millimeters, and these organisms are too small to be eaten by the salamanders, which was why they ended up on the sticky traps rather than ingested by the salamanders. And the total number of invertebrates were always greater in areas without salamanders as compared to the areas with salamanders, whether they were collected by sticky traps, mesh bags, humus, or leaf litter. The calculations from the G-test supports this difference between areas with or without salamanders. And I have a graph. This graph shows the mean number of invertebrates collected with or without salamanders. And over here we have, on the y-axis is the number of invertebrates collected through these various traps, whether the sticky traps, the leaf litter, the hummus, or the mesh bags. And the black bars show the areas without salamanders, and the gray shows the areas with salamanders. And you can see that it is consistent that the area without salamanders constantly has more invertebrates than the areas with salamanders. Okay, and the enclosures with salamanders contained significantly less of all of these invertebrates: the mollusks, the millipedes, the diptera, the Calyptera larvae, and the spiders. And the enclosures with 6 salamanders all had contained no invertebrates between 5 and 7 millimeters from leaf litter or hummus. And this is approximately the width of a salamander's mouth, so they would be more ideal for salamanders to ingest, which was why they weren't found. Also, no fly adults, ants, isopods, or crickets were recovered from the leaf litter or the hummus. And in the enclosure with no salamanders, the mean width of invertebrates was 2.5 millimeters. And in the enclosures with 2 and 6 salamanders, the mean width of invertebrates was 2.2 millimeters, which this could show a preference for larger prey in the salamanders. And there was no difference in length was observed, so the salamanders don't differentiate between length of the invertebrates. And finally, it was determined that the mean rate of decomposition in 0 salamander treatments was 17% greater than in the 2 salamander enclosure, and 11% greater than in the 6 salamander enclosure. So, we have a graph to show that. Right, this is a graph of the quantity of beech leaf litter lost from mesh bags. On the y-axis, we have the grams of leaf litter that was lost, and on the x-axis we have the enclosure with 0 salamanders, the enclosure with 2 salamanders, and the enclosure with 6 salamanders. And as you can see from the graph, the enclosure with 0 salamanders had a lot more leaf litter loss than any of the other ones. And Kim will expand on that in her discussion.
Okay, the first thing that you should notice is that the invertebrate population in the enclosures with salamanders did in fact go down. And you probably did not need to have a study to show that salamanders are going to eat the prey that they're supposed to. So that was not a huge shock to Wyman. He knew that was going to happen. But what was interesting to note is how the invertebrate population changed and how the invertebrates would affect the decomposition rate of the ecosystem and the carbon cycle of that ecosystem. And first of all, the invertebrate population. You've seen before that the average width of the invertebrates went down in the enclosures with the salamanders. And so you could conclude that the salamanders are going to be eating the larger invertebrates before they start eating any other invertebrates. And this is most likely due to the fact that salamanders are going to make hunting worth it. So they're going to go after the big prey before the smaller prey. And what's interesting to note is that the enclosures with the 6 salamanders did not have any flying ants or any crickets. So the population was so dense in that area that they ate out an entire species of crickets and other invertebrates. So that proves that the salamanders can greatly affect the populations of invertebrates in the ecosystem, since 6 salamanders could eat out all the crickets and flying ants in just one enclosure. Okay, and when the salamanders are inside the enclosure, the decomposition rate inside that enclosure would decrease significantly. And you're probably wondering how that happened, because salamanders don't do anything with the leaf litter. They don't eat it, they don't do anything with it, so you're probably wondering how that happened. And Wyman hypothesized that this was due to the fact that salamanders are eating all the invertebrates that decompose the leaf litter in that enclosure. So as the salamanders are eating the invertebrates, less and less invertebrates are able to decompose the leaf litter that are left inside that environment. So Wyman's study helped prove that salamanders can affect the decomposition rate of the ecosystem they are in. And when you decrease the decomposition rate of your ecosystem, you will affect the carbon cycle, because as you can see here, decomposition is a major part of the carbon cycle itself. They're decomposing the leaf litter, which will turn in the carbon that is from those leaves and anything else in those leaves into carbon dioxide in the air, which will infect the environment around them. And since the invertebrates are not releasing as much carbon dioxide into the air, there's going to help affect the global climate change. And Wyman concluded that an 11 to 17% reduction in decomposition could result in 261 to 476 kilograms of carbon per hectare not being released into the air. So that means there is a lot of carbon being left into the ground in a very small area. And this is going to affect the global climate change itself. And the global carbon budget is currently unbalanced, with known sources of carbon dioxide being greater than known sinks by 0.4 to 4 GT of carbon per year. And Wyman concluded that if the surface area of New York State had a significant population of salamanders, that as much as 0.4 GT of carbon dioxide would remain inside the leaf litter. And rough calculations would suggest that the carbon retained by terrestrial ecosystems with a 10% reduction in decomposition may be as high as 10 GT. And what's interesting to note is that an enclosure with only 2 salamanders could see a decomposition decrease of 11 to 17% itself. So since having no salamanders in an environment would increase the decomposition rate, it would therefore increase the amount of carbon dioxide being released into the air. So having salamanders present in your environment will help with your global climate change by reducing the amount of carbon dioxide that is being released into the air by decomposition. So in conclusion, Wyman's studies show that salamanders affect the invertebrate population by eating the larger invertebrate first. The invertebrates will not be able to decompose as much leaf litter, which would decrease the decomposition rates of that environment. And by decreasing the decomposition rate of the environment, you are going to decrease the amount of carbon dioxide that is being released into the air, and therefore helping decrease the global climate change we are experiencing today.
Thank you very much for listening to our presentation, and we hope that you gained a better understanding of the detritivore food web and the carbon cycle in regards to salamanders as a top-down effect predator. Thank you.